Right now, there are thousands of people around the world struggling for their lives. They've suffered strokes or aneurysms or have experienced severe head trauma from terrible accidents. A week ago, a few days ago, even a few hours ago, they were going about their daily lives. But brain injury can strike in the blink of an eye, and when it does, lives can be changed forever. But what exactly is happening in the brain as patients with serious injury lay unconscious for hours or days, struggling to regain function? That's a question that has puzzled doctors for decades. In recent years, however, a handful of scientists around the world have learned to peer into the black box of the injured brain. They've discovered a surprising process that continues to impair brain function for days and even weeks after stroke and traumatic brain injury, raising new hope for a breakthrough to treat these devastating injuries. This is their story. In the 1940s at Harvard University, a graduate student was working on his PhD thesis to learn more about epilepsy. Aristides Liao was recording electrical signals from the brains of rabbits and saw something that no one had seen before. He put electrodes across the surface of the brain and started to record the electrical signals. He stimulated a point near the first electrode and saw a depressed signal. This depression spread to the next electrode, and the next, and the next. He then saw the activity in the first electrode return back to normal, and then in the second and the third, until all were restored. What Liao saw was a wave of depressed activity spreading over the surface of the brain. This odd wave of brain dysfunction immediately seemed to be the pathophysiologic basis of at least one neurologic condition, the migraine aura. Approximately 1/3 of people who suffer migraine headaches experience profound neurological disturbances known as aura that precede the headache phase. These disturbances, most often occurring in vision, have been famously depicted by artists who suffer migraine, showing the loss of perception that begins as a point and spreads across the visual field in a wave or spiral pattern. The propagation of these waves was studied scientifically by the neuropsychologist Carl Lashley, a contemporary of Aristides Liao. Remarkably, the speed of the moving visual deficit precisely matched the speed of Liao's spreading depression wave. Following these breakthroughs in the 1940s, for the next 60 years, it was thought that Liao's spreading depression was relevant in neurology mainly as the brain mechanism of migraine aura. In the 1980s and 90s, however, there was a new breakthrough as scientists began to observe these same waves in experimental studies of stroke. In London, a neurosurgeon at King's College Hospital was one of those scientists.
Well, in the mid-70s, when I was towards the end of my neurosurgical training at the National Hospital in Queen's Square in London, I had the good fortune to work for a year or so as a research fellow with Professor Lindsay Simon, who was doing a lot of work at that time with the experimental stroke model in primates. And around about that time, we got access to ion-selective electrode technology, particularly potassium microelectrodes, and we started placing these in the penumbra. And in fact, the term penumbra derived from the finding that, in fact, electrophysiological failure happened at higher levels of perfusion than did membrane failure. And it was only when we got down to really quite low levels of flow that you got sustained increases in potassium in the extracellular space. So the message was that, in fact, loss of electrical function was not due to membrane failure, but something much more selective, and only when the flow got lower did you get complete depolarization. So this was the origin of the term penumbra, and it actually had quite a bit of a hand in developing this term and inventing it, really. And we began to find that just as in the potassium electrode experiments in primates with Lindsay Simon, what we were beginning to see was the recurrent spontaneous increases in extracellular potassium. And we'd become aware in the late 70s with Neil Branston in Lindsay's lab that other people had doing similar sort of studies on spreading depression and seeing these same changes. So we came round to the idea, particularly Neil Branston and I, that these potassium transients that we were seeing were actually manifestations of spreading depression.
Scientists then began documenting how spreading depression waves cause further brain damage after stroke, trauma, and ruptured aneurysms. But this line of research never gained a strong foothold in biomedical research since there was no evidence that spreading depression occurred in the human brain. In fact, the consensus in the biomedical community was that the human brain was too highly evolved to be vulnerable to such a profound dysfunction. It took a neurosurgeon with the right scientific background, using the right techniques, to change that thinking.
So I was very lucky. We moved into quite close collaboration with an epilepsy surgeon who I have come to know very well over the years, Professor Charles Polke, and I asked him about the feasibility of putting subdural multi-contact electrode strips on the brain surface at the end of an operation. And he said, it was very reassuring, he said, yes, we do this the whole time for patients with epilepsy who are candidates for neurosurgery. And so we started to do that. And the very first case we did, we saw series of these events which exactly replicated what was published on Leon's original paper in 1944. So that was really quite exciting to us, and when we first got these traces, I thought, well, we better get some further opinions on this. So I contacted Martin Lauritsen and Martin Fabricius in Copenhagen and sent them the data that we got. And in fact, they got on the next plane virtually from Copenhagen, and we spent a very exciting day looking at these traces and realized that this was something rather special and quite exciting.
Dr. Martin Lauritsen, a professor of clinical neuropsychology at Copenhagen University Hospital, recalls this collaboration.
Suddenly in the beginning of 2000, I got some emails from Tony, because he had now a new idea about how to record spreading depression in humans. And these emails and the following telephone correspondences became of the start of the COSPID project. So in Copenhagen, I was working with Martin Fabricius, another COSPIdian, who is an expert of electrophysiology like myself. And we had a lot of interaction with Tony, designing how to record spreading depressions in humans, and also in the interpretation of the data. And it was a very happy day that after a lot of footwork, Tony made the first recording of spreading depression in a patient in King's College Hospital. And we got a mail, I think, or an express mail from Tony with the files, and could see that this was in fact a spreading depression.
Jed Hardings, a neuroscientist at the University of Cincinnati, is one of the many researchers who were inspired by this breakthrough. He explains the current understanding of what happens to the brain during spreading depression or spreading depolarization.
You see, normally, brain cells have stored electrochemical energy. They have an electrical polarity similar to a battery, and it's this stored energy that drives electrical function. And when a spreading depression occurs, cells lose this polarity. In other words, they become depolarized. The batteries are drained, and so the cells lose their function. And this is why we now refer to spreading depressions as spreading depolarizations, to describe the essential mechanism of dysfunction.
More colloquially, spreading depolarizations are sometimes known as brain tsunamis.
The EEG basis for measuring spreading depolarizations might also imply that these are purely electrical events. But when you consider that the entire physiologic and anatomic structure of the brain from membrane proteins to metabolic pathways to vascular architecture, is designed to support electrochemical membrane potentials and signaling, then the catastrophic failure of collapse of those membrane potentials during spreading depolarization necessarily implies profound consequences for all aspects of brain physiology. Spreading depolarizations are indeed brain tsunamis, and the analogy is appropriate because everything is affected in the wake of a spreading depolarization. Morphologically, cells swell, dendrites become beaded, synaptic spines retract. The brain tissue becomes acidic, neurovascular coupling is disrupted, blood flow changes, and energy reserves are depleted. Even on the molecular genetic level, the profile of gene expression is completely changed.
The effect of spreading depolarizations to restrict blood flow in the brain was first discovered by Dr. Jens Dreyer, a neurologist at the Charité Medical University in Berlin, Germany. He further explains the breakdown of electrochemical balance in the brain during spreading depolarization and its cost to the brain's energy supply.
So to summarize this part, a neuron is a power station in which chemical energy is produced in the form of ATP. The ATP fuels the sodium pump, which creates concentration gradients of small ions across the cellular membrane. These concentration gradients store a large amount of electrochemical energy. Only a tiny amount of this electrochemical energy is spent during a neuronal signal. In other words, neurons follow Merkel policy. They are very rich in electrochemical energy and they spend only tiny amounts of this energy for signals. So they have $1 million in stock and one signal costs them 50 cents. In principle, there are only 3 pathological situations in which neurons suddenly release large amounts of the energy contained in their ion gradients across the membrane. An epileptic seizure costs them about $130,000. There's another phenomenon called spreading depolarization, which costs them about $800,000, and death costs them about $1 million.
Thus, spreading depolarizations induce a twilight state of near death for brain tissue. The degree of disturbance can be appreciated in recordings of these pathologic states, comparing the small deviation from normal brain homeostasis during seizures to the relatively large deviation during spreading depolarization. The latter, in fact, is electrically equivalent to the state of death, and if it persists long enough, will cause irreversible brain damage. This terminal spreading depolarization, as observed after stroke or cardiac arrest, was also first discovered by Liao in the 1940s. Dr. Strong's contribution was not only discovering these events after injury to the human brain, but also providing the medical community with a method to monitor spreading depolarizations that could be easily adopted at other medical centers. Let's take a look. The process starts by placing an electrode strip on the surface of the brain. The amp must be capable of recording frequencies below the normal EEG range, at least down to 0.01 Hz or even 0.0 Hz, known as DC coupling. When displaying the EEG, a compressed timescale is required, at least 30 minutes per page. To show the large depolarizations, a display scale of up to 5,000 microvolts peak to peak and a lower cutoff filter of 0.01 Hz or less is needed. To show the EEG depressions, a display scale of 1,000 microvolts peak to peak and normal EEG filters is typically used. Following Dr. Strong's initial publication in 2002, a group of investigators from different hospitals gathered in Copenhagen and formed a consortium, the Cooperative Studies on Brain Injury Depolarizations, or COSBID, to build on his finding. In the next years, more findings were published that established the relevance of spreading depolarizations to patients' recovery from stroke and brain trauma, and adoption of this monitoring grew. Today, more than 18 neurosurgical centers in the U.S. and Europe are monitoring spreading depolarizations in patients, and more are joining each year. Dr. Oliver Sakowitz, a neurosurgeon at the University of Heidelberg, was one of the first physicians to begin monitoring spreading depolarizations in stroke patients, and now does so as a routine procedure in managing patients.
I was not so sure about spreading depression right from the beginning and the occurrence in the human brain. It was then when we looked at our even noisy recordings closer that we were actually lucky, even with those, to find spreading depolarizations, because the signal was so much larger than any of the noise that we captured that we actually found spreading depolarizations even in those recordings. When it happened that we were looking at individual patients where we saw spreading depolarizations occurring at high numbers, and they suddenly stopped in the recordings, and we went back through the records looking at the different interventions that were taken, looking at the different pharmacological agents that were added, when we found that in these particular patients where a train of spreading depolarizations suddenly ended was when they were started on ketamine, which is a sedative drug that we add on to our sedative regimen whenever we don't get along with the standard sedatives. Then we add ketamine, and we saw right at that time spreading depolarization stopped. So that was something where I thought, well, despite all skepticism, what would that be an artifact that can be stopped by a systemically given agent right away? So we went through all the literature again, and obviously we were not the first to describe ketamine as an agent that is able to stop spreading depolarization. So it was not a new finding, but it was the first time that we actually saw this in man, and it was the first time that I was turned from a skeptic to a believer.
The striking nature of these data further stimulated Dr. Sakowitz to pursue experimental studies of how spreading depolarizations propagate.
One additional thing we have been looking at in our lab is the way spreading depolarizations travel across the brain surface. And what we were particularly looking at was a gyrated surface of the mammalian brain. You know, much of the studies that have been done in brain research are based on rodent models. And rodents, they have a flat, so-called lissencephalic brain. And the spread across the brain surface may quite differ from the spread that we see along a gyrated human brain, where not all waves travel the same distance. They can be stopped by barriers like brain vessels, fissures, and sulci. And the model that we've been using is a pig, which actually we do not have much in common with pigs, but what we do have in common is the gyrated brain, and the mechanisms that spreading depolarizations travel across the brain surface may be quite similar to what we see in the human brain. Here in this small video sequence, you see the exposed hemisphere of a pig. And what we do here is we make use of so-called intrinsic optical signal imaging, which essentially tells us the amount of blood volume in the brain during the course of an event. And you see here, now that we stimulate it with a pipette, with a small potassium droplet, you see the start of a spreading depolarization wave emanating on one gyrus, and you see a number of 5 cycles now of this wave cycling around these 2 gyri, and this video covers more than 2 hours and is sped up for better visualization. And as you can see, this type of reverberating wave is something that in electrocorticography readings would turn out like a cluster of spreading depolarization events repeating in a cyclic fashion.
Dr. Raymond Hellbach, a neurointensivist at the University Medical Center in Innsbruck, Austria, took an interest in spreading depolarizations after reading the emerging new medical literature.
Hello everybody, my name is Raymond Hellbach. I'm a neurologist and neurointensivist, and I'm working in the neuro ICU here in Innsbruck in Austria. As you can see in the background, we have a lot of mountains here, a lot of skiing accidents, so we are referral center for trauma. We also have a lot of patients with intracerebral bleeding and ischemic stroke, which are usually transferred to our ICU. And you know, as always, when you start something new, there are some problems at the beginning, but with the support of COSPIT members, we actually didn't run into severe problems in terms of recording, and had very interesting experiences with even the first patients. And we recognized these clusters of cortical spreading depolarizations in our patients, and also recognized that certain episodes of brain tissue hypoxia, which were not explained by perfusion or metabolism, were very much closely related to clusters of cortical spreading depolarizations. So COSPIT monitoring is still considered as a research tool in our unit. However, it more and more gets into clinical practice, meaning that when we round on our patients, this is a valuable additional information whether the patient had additional clusters of cortical spreading depolarizations in the last 24 hours. What we consider is that clusters of cortical spreading depolarizations may really contribute to secondary brain injury.
As said, my role in neurointensive care has been multimodality monitoring, and we have worked with many different types of monitors, coming from microdialysis over PTiO2, ICP, CPP monitoring, EEG. And I think this particular spreading depolarization research gives us a 1 or 0 answer if secondary injury is happening at the time of monitoring. So more than looking for thresholds in the different type of monitors, you here have a clear answer whether something bad is ongoing in the injured brain and in our comatose patients.
Now, we have been following stroke patients for centuries. We have been testing them, we have been asking them what they experience. And it's a very rare thing to have a stroke patient tell you that they have something like an aura, like a scintillation scotoma or some march of sensory things crawling up their arms, which is what we would usually look for if we compare to the migraine aura, which is the same basic pathophysiological phenomenon. But in reality, if we go to the patients with acute vascular damage to the brain, we can find these episodes, but they occur in various areas of the brain. So one example was a lady who had suffered a subarachnoid hemorrhage, and she had recovered reasonably well after having her aneurysm clipped, and we were monitoring her for several days. And then after a few days, I went to her bedside, talked to her. She was alert, she was oriented. I went to the nurse's room, I talked to them, gave instructions, and 20 minutes after I came back to the patient, who was now obtunded, aphasic, and I could immediately see on the strip recording that since I left her, there had been an episode of spreading depression propagating through her left perisylvian area.
Dr. Dreyer further discusses the utility of spreading depolarization monitoring.
Yeah, I think the monitoring of spreading depolarizations in patients has a great potential because when patients are analogous sedated, when they are ventilated, it's very hard to assess them neurologically, and it's very difficult actually to find out what's going on in my patient. So if you have a method that tells you this patient now develops a brain infarct, then you can interfere with the situation and you can treat the patient, because with brain ischemia or stroke, time is brain. So if you are too late with your intervention, if you, for example, first have to bring the patient to a scanner to find out that something is developing, then you lose too much time. And therefore, this monitoring is extremely valuable. It might give you hints, patients developing a stroke, patients developing other complications like a hypoglycemia, a septic condition, and many other things. I mean, we use this principle of electrophysiological monitoring already for a long time for the heart with the ECG, which you find on any intensive care unit, and we need such a real-time monitoring also for the human brain, and not only for the other organs. So that is one potential, and then of course the mechanism itself is very interesting, and we would like to interfere with it.
So in the end, it's a bit curious that following Leal's first publications in the 1940s, that it was the spreading depression theory of migraine that took hold and dominated thinking in this field for so many years. Because really in those publications, he also showed the connection between brain injury and spreading depression. And although I don't think he quite realized it at the time, in retrospect, he really was the first to show that spreading depression, spreading depolarizations are the final executioner leading to brain death. So it's gratifying that finally in these recent years, we've been able to catch up with his theories and begin to translate them into clinical practice when caring for patients with serious brain injury. It's a very exciting time, but I think we are still only at the beginning.